A depyrogenated tunnel is a piece of equipment used in sterile pharmaceutical manufacturing to remove pyrogens, particularly bacterial endotoxins, from containers such as vials or ampules. It operates by exposing the containers to high temperatures, typically around 250 degrees Celsius to 300 degrees Celsius, for a specified period, effectively destroying any endotoxins present on their surfaces. The tunnel is part of an automated line that usually follows the washing stage, where the containers are cleaned and prepared for sterilization. Once in the tunnel, the containers move through different zones: a heating zone, for depyrogenation, a holding zone, to maintain the high temperature for the necessary time, and a cooling zone, to reduce the temperature before the containers exit the tunnel. Depyrogenation tunnels are essential in ensuring that containers used in sterile pharmaceutical products are free of harmful pyrogens, which could lead to adverse reactions in patients, such as fever or more severe complications. Validation of the depyrogenation process is critical, involving biological indicators like endotoxin challenge testing to ensure that the tunnel meets regulatory requirements for endotoxin removal. The operation of a depyrogenation tunnel involves several key stages that ensure the removal of pyrogens, specifically bacterial endotoxins, from pharmaceutical containers like vials or ampules. Here's an overview of the process. Loading stage. Washed containers, vials, ampules, are loaded onto a conveyor belt system after going through a washing machine. The containers are typically made of glass and should be free of visible particles before entering the tunnel. The containers are evenly spaced on the conveyor belt to ensure uniform heating. Heating zone, hot zone. The containers pass through the preheating zone, where their temperature begins to rise gradually. This prevents thermal shock when entering the main heating area. The containers then enter the main heating zone, also known as the depyrogenation zone, where they are exposed to extremely high temperatures, typically ranging from 250 degrees Celsius to 300 degrees Celsius, for a defined period. The heat effectively destroys pyrogens, particularly bacterial endotoxins. The temperature and residence time in this zone are critical and must be validated to meet regulatory standards, typically achieving a 3-log reduction in endotoxin levels, commonly considered adequate depyrogenation. Holding zone. In the holding zone, the containers are maintained at the high depyrogenation temperature for a specific period, ensuring sufficient heat exposure to destroy endotoxins. The holding time is critical to ensure complete depyrogenation. This is based on the heat penetration validation study, confirming that even the coldest spot on the vial or ampule reaches the target temperature for an adequate duration. Cooling zone. After depyrogenation, the containers move into the cooling zone, where their temperature is gradually reduced. Forced cooling mechanisms are often used, like filtered air, to bring the temperature down to a level suitable for handling, while maintaining a sterile environment. Unloading stage. Once the containers are cooled, they are unloaded from the depyrogenation tunnel and transferred to the next step in the manufacturing process, usually sterile filling under aseptic conditions. Control and monitoring. The entire operation of the depyrogenation tunnel is controlled by a programmable logic controller, PLC, which ensures that all operational parameters such as temperature, conveyor speed, and airflows are within set limits. Continuous temperature monitoring with sensors, thermocouples, ensures that the process meets the defined specifications. The system must have alarms to notify operators if any deviation from the set parameters occurs. Validation and qualification. Regular thermal mapping is required to ensure uniform heat distribution across the tunnel. Endotoxin challenge testing is performed using endotoxin indicator vials to confirm the tunnel's effectiveness in pyrogen removal. IQ, OQ, PQ, installation, operational, and performance qualification steps are performed for proper validation of the tunnel before use in production. Proper operation and validation of the depyrogenation tunnel are essential to ensure compliance with regulatory standards like CGMP and to avoid contamination in sterile pharmaceutical products.